Hello class. Today we're going to go ahead and finish our talk about metabolism. Okay, so last time we finished off when we were talking about exergonic and endergonic reactions. Okay, so today we're going to start talking about how enzymes help with the metabolism. All of an organism's chemical reactions are due to the help of enzymes. Okay, each step is actually catalyzed by specific enzymes. Enzymes are very, very specific on what kind of substrate they're going to react upon. So right here, from this green looking substrate, enzyme A will react and use it up and break it down and change it into this blue number 2 substrate. Then this substrate will undergo a reaction with the enzyme B and become a whole different product right here in the next one, which would be this brown color. Then this brownish number 3 will actually be used up and basically be broken apart or transformed with enzyme C. And that is your final resulting product, which is like this nice bright orangish color. So every single step is catalyzed by a very specific enzyme. So catalysis' definition is a process where an enzyme makes a specific reaction occur much faster than it would normally. So the reaction will occur without the enzymes there. They just help speed up the process quite a bit. Okay. Even after an enzyme is used up, it is not just die and go away. It actually is reusable. So it's reused over and over and over again. Most enzymes are proteins, and a substrate, which is what we're looking at down here, this green marking right here, this green substrate right here, that's a substrate. It's a specific reactant. Remember, reactants are at the beginning of our chemical reactions. It's a specific reactant acted upon by an enzyme. So at the very end, even without the enzymes, we would still get these two products, but this helps it happen a lot quicker. So here's our starting substrate. Here's our enzyme. Okay, it's very specific. You see how it's perfectly shaped to attach to this actual enzyme right here? So this substrate and this enzyme are going to become an enzyme substrate complex. Okay, in doing so, when they actually attach, there's actually a slight change, okay, that actually occurs. It's an enzyme that's going to have a slight conformational change. And by the way, the point that you would call where this substrate attaches to this enzyme is the active site. Okay, so they're attached right here. There's a slight conformational change in the enzyme, and that also results in the new two products. So we went from a much larger substrate or reactant, and it's undergoing its change with the enzyme helping it along. And last but not least, we end up with two new products at the end here. So how do active sites work? Okay, at that point where the substrate is attaching to the enzyme, they orient the substrates correctly. They stress the substrate bonds. It provides a favorable microenvironment for that substrate and enzyme, and it's going to direct the participation of the active site in the chemical reaction. Okay, substrates converted to products at this active site, and then at the end of the whole chemical reaction, the products are finally released. So let's go ahead and look back at our endergonic, exergonic reaction graphs and kind of put an enzyme into play here. So remember we looked at these graphs when we were talking about endergonic, exergonic. So let's go ahead and have a recap really quickly. This is our reactants. These are our products up here. So as you can see, there's been an increase in energy. That means there's been an input of energy. Well, input of energy means that it is an endergonic reaction or an anabolic reaction. Okay, so activation energy. What are we talking about when we talk about that? That is the amount of energy that is required for any reaction to begin undergoing that chemical transformation. So what happens here? The purple line you're seeing right here, that's the activation energy actually required to start this reaction. Once this activation energy is reached, then the action can actually recur and result in our products. So this is actually happening normally, the purple one. But if we add an enzyme in there, it is actually going to be reducing the amount of activation energy required for the reaction to occur. So imagine how much energy you would need if this was a boulder. You had to push up this tall, giant hill. before it actually came crashing down and broken into smaller rocks, which is what you want. So let's say now you have an enzyme help you out. So guess what? You don't have to do as much work. You only go up to right here. Then you can go ahead and drop that folder off. So enzymes do not change, as you can see, they do not change the effect in the delta G, in the free energy, or the result in the products. It only changes and it lowers the amount of activation energy required for the reaction to occur. So back in the day, they used to think that enzymes were actually a locking key mechanism. In other words, they fit perfectly together. You put a key in a lock, you can turn it, you're great. Okay. Later on, we actually found out that it's not a perfect fit. It's not a lock and key mechanism. It's actually what they call an induced fit. So remember, at the very beginning of this lecture, I stated that right here, where the enzyme substrate complex are united, where these guys are actually one, there is a slight conformational change in the enzyme's shape. That just makes it actually shape a lot better with that substrate. That is what they mean by induced fit. In other words, this is not a perfect fit when they get together, but after they actually combine and attach, then we have that change in the enzyme. After that change occurs and allows for that great binding site, then we're going to have the chemical reaction occur and then the products be released.
Making a snowman is always a big ball of fun. 3 snowballs full of fun to be exact. And each snowball starts out small, getting bigger as you roll it in white fluffiness. Let's imagine that you go outside in a fresh snowfall to build one of these guys, but you don't have the best suited yard for rolling snowballs. In fact, your backyard is one giant hill. This hill is fantastic for sledding, but not so much for making snowmen. Anyways, in order to make a giant snowball for the snowman's bottom, you'll need to actually push this snowball up over the hill and then down the other side. You do it because it's fun, but make no mistake, it takes a lot of energy to get started up this hill, and this is hard work. Right now, you share a quintessential problem with some chemical reactions within a cell. These chemical reactions might be a lot of fun when you're in a cell, but it takes a lot of energy to get them started. This is called the activation energy, or the energy required for a reaction to start. You can think of activation energy as that energy you needed to muster up in order to push that snowball up and over the hill in your backyard, in order to maximize the size of your snowball and get it to the other side. What your cells have to help overcome a problem of high activation energy are called enzymes. Enzymes are proteins that lower the activation energy of a reaction. In doing this, enzymes increase the rate of a reaction, helping it to occur faster. However, enzymes are not consumed in a reaction, they simply help it to occur. They do not add snow to your snowball, they just make it easier to make a snowball. This makes enzymes reaction catalysts. The presence of enzymes is analogous to decreasing the size of your hill in your backyard. Enzymes make things easier for your cell and help chemical reactions occur. There are hundreds of different kinds of enzymes in your cells, which all participate in different types of reactions. Enzymes can break molecules apart, build or add molecules, and even rearrange them. In lowering the activation energy of a reaction, enzymes decrease the barrier to starting a reaction. It's important to note, however, that the change in energy remains the same between the start and end of a chemical reaction. For example, if a reaction released 200 kilojoules of energy without an enzyme, the same reaction would still release 200 kilojoules of energy with some enzymatic aid.
So that was a great informational video. Okay, kind of made it very simplistic as to what's going on with the enzymes. And that video is actually embedded in the notes, so you can just click on it if you'd like to watch it again. Now, enzymes are very specific as to what they will work on and what they do, okay? Number one, they're very specific for what substrate they're going to attach to, okay? Also, since they're very specific on that substrate, they're going to form a very specific enzyme substrate complex. They have an active site formed only to attach to that one or two substrates, whatever ones they actually want to react upon. And cofactors can also influence how specific an enzyme is going to react or not. And competitive and non-competitive inhibitors will also affect the specificity of enzymes. And let's go ahead and take a look at each of these. So let's look at cofactors first. Okay, cofactors are required by many enzymes. Cofactors are non-protein enzyme helpers. Coenzymes are also organic cofactors. So what does that mean? They are actually helper molecules that assist in biochemical transformations. So you can basically imagine these ions that are labeled right here, or these atoms, are actually meant to sit there and actually be a helper molecule for the enzymes to do its job it was already going to do. Right here, let's go ahead and start talking code. So we already know we have enzymes that react upon chemical reactions. They reduce the amount of activation energy required for a reaction to occur. They can have helper molecules known as cofactors. And let's figure out what else can affect an enzyme's activity. Number one, an enzyme's activity can be affected by temperature. It can also be affected by pH levels. It can also be affected by the salinity level and cofactors available or not available for it. And also, last but not least, sorry, went by too fast, it can also be affected by the inhibitors that are present at that time that it is trying to react with that substrate. So, temperature. How can temperature affect the activity of an enzyme? So, all enzymes actually have an optimal range that they perform the best in. So, a temperature that they like the best. They also have an optimal pH range and a salinity range. So, if we start manipulating these ranges, it'll change the enzyme's activity. So let's go ahead and see what happens when we increase the temperature of an enzyme. So if we increase the temperature that a chemical reaction is occurring in, here's our enzyme activity, here's our temperature. Let's look at E. coli polymerase first. And remember, if it ends in ASE, it is an enzyme. So we have two enzymes here. We have E. coli polymerase, what we're going to look at first. If we have this chemical reaction occurring at a 20 degrees Celsius, the enzyme activity is very, very slow. But if we actually increase the temperature to 40 degrees Celsius, which seems that this is the enzyme's optimal temperature range because it's actually performing at its best here, then there is going to be a lot more enzyme activity occurring. That means a lot more chemical reactions occurring because of these enzymes helping them out. If we actually put the temperature a little too high, you can see that this graph does not continue on. It actually stops here around 96 degrees Celsius because if the temperature ends up being too high, it will denature the enzyme, or it's actually changing the shape of the enzyme. So instead of it being a perfectly perfect shape with a triangular active site, let's say it changed the conformational form of that enzyme into, let's say, a square shape instead. That's called denaturing enzymes, which means it is no longer functional. It cannot be used anymore if you go too high of a temperature with that enzyme. Okay, now let's look at optimal pH range. So optimal pH, if it's too acidic, meaning pH of 2, pH of 3, it can actually denature an enzyme, or if it's too basic. So if it's too high, let's say pH of 14, it'll denature the enzyme as well. So the optimal range for this pepsin would be around 2 pH, so it's very, very acidic. It actually likes acidity. But if we start getting more along the neutral levels of pH, it actually completely denatures it at around 6 pH. Trypsin, on the other hand, has an optimal pH of 8. So if we had a chemical reaction undergoing and we changed the pH to 8, trypsin would be actually going through these chemical reactions a lot quicker and faster because this is its optimal pH range. But if we put it in a pH of, let's say, 3, it would be denatured and it would no longer function because of that 3D change in the protein. Same thing over here. If we went too high, it's going to denature that enzyme as well. Salt levels also affect the hydrogen bonds because those are the ones that are holding those enzymes in their 3D shapes. So I know we talked about inhibition or competitive and non-competitive at the beginning of this lecture. So now we're going to find out what that means. So an inhibitor is just like it sounds. It's going to inhibit or stop the action of an enzyme one way or the other. So first type is going to be competitive inhibition. That just means that, let's say this is the enzyme, so naturally, just like the other picture we saw, we would have a substrate come and try to attach at this active site, which this is the substrate right here trying to come over here. But since it's competitive inhibitor that's around, which is it's inhibiting the substrate and the enzyme from coming together, okay, it's going to prohibit it, it's going to stop it. This competitive inhibitor is going to come and attach to the active site instead, so it can actually block that active site and the substrate can no longer attach. That's why it's called competitive, because it's actually competing with the substrate to get there before it can, to prevent it from undergoing and doing its chemical reaction. This can be temporary or a permanent effect. Non-competitive inhibition, on the other hand, is when an inhibitor comes along and it's not competing for that active site on the enzyme. It's actually going to attach to the enzyme on a different location. This is the location called the allosteric site. When an inhibitor attaches to that allosteric site, it's going to change the shape of the enzyme. It's going to change that conformational shape of the enzyme, okay? So if this enzyme is changed, guess what? That substrate that was once able to bind can no longer bind, and it just kind of goes away because now it cannot undergo its chemical reaction with the help of an enzyme. That's what this inhibitor does. That's an allosteric inhibition because of where it's sitting here. It's attaching to the site other than the active site, so that's the allosteric site, and it's still inhibiting or prohibiting this enzyme substrate complex from coming together. Allosteric activation is the same kind of attachment. We have the activator come and attach the allosteric site. And remember, I'm saying activator now, not inhibitor. An activator comes and attaches to this allosteric site, and look at this active site over here. It was not able to actually attach to this substrate yet. But when this activator comes and attaches to the enzyme, it changes the shape of the enzyme, making it a much better fit for this substrate right here. So this is allosteric because of where it attaches. Both of these are allosteric because it's a site other than the active site. Allosteric inhibition, though, prevents the substrate complex and the enzyme coming together. And allosteric activation, will attach an allosteric site, but it will change it and actually allow a better fit for the enzyme substrate complex. Sometimes we have what's known as feedback inhibition. This is when the end product of a metabolic pathway shuts down the entire pathway. So let's go ahead and go through this from the beginning to end. We're going to go ahead and start here with the substrate. So the substrate is this green arrow. It's going to come over here. It attaches to this enzyme. They combined create the enzyme substrate complex. Then that actually undergoes its conformational change. So this enzyme is changing this triangle into this long purple rectangle. Then this purple rectangle is very specific for this enzyme. They attach, make a substrate enzyme complex, and that results in this blue kind of wavy looking thing. Okay, this is the product of that reaction. So now this is the reactant, and substrate is going to come attach to this enzyme. They once again form another enzyme substrate complex, and that results in this final end product. Well, this end product comes around, and guess what it's going to do now? It's going to attach to this allosteric site of this enzyme, and that changes the shape of this enzyme. So once the enzyme shape was like this, and it was actually very good to connect and actually react with this substrate, but now the end product came back and changed its conformational shape so that way the substrate is no longer able to actually attach to this enzyme. That is what's known as feedback inhibition. It's when a product itself can be a regulator. So I know we talked about cofactors previously, but these are going to be the real definitions of what a cofactor and coenzymes are. These are going to be the helper molecules, remember? So they're atoms or molecules other than proteins that are necessary for an enzyme's function. Coenzymes are organic cofactors such as vitamins. So how can an enzyme affect you with something that you can see? Since we don't actually get to see all of our awesome cells doing the cellular respiration and breaking down those glucose molecules to release our energy, we don't see all that. So let me give you an example that you actually physically can see. Some of you have heard of the brown recluse spider. It actually has an enzyme-based venom. Remember, enzymes are used as catalysts. They actually are used to help chemical reactions occur a lot quicker. So, the enzyme that this brown recluse spider has is sphingomyelinase D. It actually causes necrosis, or it's known as tissue death. How it does that is, you know how we have the plasma membranes around our cells to protect our DNA and everything else in our cells? Well, guess what? This guy with that enzyme is able to destroy our cell membranes, and that's what's causing the tissue death. So right here, you can actually see the spider bite after a couple of days. It's actually been allowing those reactions to occur a lot quicker than they would have normally, because remember, it's an enzyme that this spider has in its venom. So it's actually able to break down our cells membrane very, very rapidly. So this has been a couple of days. And this right here is unfortunately a woman who actually saw the spider on her foot as she got out of the shower. So obviously the spider was coming from the drain itself. And eventually she had to have her leg amputated below the knee due to this necrosis that was occurring. It's really sad. So if you ever get bit and you're unaware of what type of spider or anything it is, just obviously always go to the doctor, because you never know. You want to catch anything like this as soon as possible so you can stop and hopefully reverse the necrosis that's occurring, or do whatever your doctor advises you to do. So let's go ahead and talk about what ATP is actually made up of. The ATP molecule is composed of three components. It is a sugar molecule right here. Okay, it also has these three phosphate groups and adenine. That's why it's called adenosine triphosphate, because of these three phosphate groups right here. So what happens is these three phosphate groups right here are actually negatively charged. Each of them are negatively charged. And as you all know, opposites attract. So since those three phosphate groups are negatively charged, opposites attract, these are not attracted to each other. So imagine like it's you and your two siblings. You're sitting in a very small seat on a school bus. You're ready to just get away from them as quickly as possible, or most siblings are. And so you're kind of just ready to jump off that seat and get into your own seat so you can be more comfortable. That's kind of what ATP is reacting like. So how we act upon that is we actually add water to this reaction, this is called hydrolysis right here. And when we add water, what that does is actually break apart that bond right here. And remember, whenever we break a bond, we release energy. So we're able to break off that one phosphate group, which ends up being an inorganic phosphate group all by itself. This becomes adenosine diphosphate, which means there's two of them, and the release of energy, which is what we wanted. Remember, ATP is really good for a quick burst of energy. So what is energy coupling? Just like you would imagine, coupling, okay, it's basically the energy from an exergonic reaction, because remember, exergonic, they have a release of energy. So that energy from that exergonic reaction of ATP hydrolysis can be used to drive an endergonic reaction. So overall, the coupled reactions are exergonic. Let's go ahead and take a look at one. Right here we have glutamic acid and ammonia. When we actually build those guys together, it's an anabolic reaction, which means we have an input of energy, and that's why it's positive delta G. An exergonic reaction though, we have ATP, remember those three phosphate groups ready to come apart, and then we add water. That results in our ADP, an extra phosphate group, and a release of energy, which is this negative 7.3 right here, this negative free energy. This is the actual energy being released from this whole system. So how does this ATP perform work? So just like we were talking about when we were talking about the coupling right now, is ATP is right here. We go ahead and undergo hydrolysis, basically adding that water, and it breaks apart that bond. and results in ADP plus a free phosphate group. And so that energy released is actually used right here by this sodium ion. The energy gets released, let's say right here, and then that's allowing the sodium to get pushed in. When you add a phosphate group to any kind of molecule, it is considered to be phosphorylated. So once we break down ATP, at the very beginning, remember we did ATP, once we break off that last phosphate group and it becomes ADP and an extra phosphate group, and the extra inorganic phosphate group, right? And we release energy. Okay, so that's what's showing on this side. This is what happens when we broke it apart. So we released energy after we broke down that bond. Energy was released for cellular work. What if we want to go back to ATP? What if we need ATP to be made again so that we can use it again? So this is what's going to happen. On this end, remember over here we had to go hydrolysis, we had to add water. So on this end we're going to actually be removing water, okay, in a form of dehydration synthesis, and energy from a catabolic reaction. Remember, catabolic, you're tearing a bond apart. Another catabolic reaction that's occurring is releasing energy into this system, and that's what we're going to use to actually build back up that from ADP and phosphate. We're going to need an input of energy, because remember, you're going from a smaller molecule, ADP and phosphate, going from a small to a much larger ATP molecule, so we need an input of energy. So that's where the energy from a catabolic reaction is where we're going to get the energy from, and that all in all results in our ATP molecule once again to be used as needed. Sucrose is broken down to glucose and fructose. So we're going from a much larger molecule, which is sucrose, and we're breaking it down with a chemical reaction into glucose and fructose. It's two smaller molecules. What kind of reaction does that look like? A catabolic or an anabolic? You said catabolic, you're correct. Catabolism, remember, is the breakdown of a larger molecule into smaller molecules. And when you break apart a bond in the larger molecule to create these other molecules, you're actually having a release of energy. Energy is getting released into the system. Okay, that's why we also have this negative delta G. So in that graph that we drew at the very beginning of free energy, this is the free energy over here, that means that our sucrose molecule would have had all this free energy, and then we ended up with our two products down here, and that's what resulted in our negative delta G, because it went from a higher energy state to a lower energy state, and that is due to the release of energy in the system. So is it spontaneous or is it not? Yes, this would be considered spontaneous because it's actually an exergonic reaction or a catabolic reaction, and both of those have the release of energy and are spontaneous. Let's go ahead and recap. Metabolic pathways, there's catabolism and anabolism. There is endergonic and exergonic reactions. There is kinetic and potential energy. We discussed what entropy is. We discussed the first and second law of thermodynamics. We discussed ATP and hydrolysis and how it looks when we're actually adding water to ATP to break it apart. And all of these are undergoing cellular regulation, and that's by manipulating the different feedback inhibitions with the enzyme activity, cofactors, coenzymes, and all these other items that come into play when we're talking about regulating the cell's metabolism.